So quickly, for the sake of time, Constanza is at the NCI in Washington and has been involved in gastric cancer epidemiology for decades. And welcome.
Thank you, Markus. Thank you, everyone, for staying here. Greetings to our online audience. Thank you to you, and Robert, for creating this protected space to foster collaboration and to promote action. It's really a pleasure to be here. I'll be discussing about biomarkers for gastric cancer. So gastric cancer is a heterogeneous disease, and there are two major efforts that documented this heterogeneity in terms of the molecular changes. We have TCGA and also the Haejin Cancer Research Group. Because of this heterogeneity, we will need multi-biomarker panels to assess and capture the heterogeneity. As you can see here, for example, from TCGA, it is estimated that 8% of the tumors are Epstein-Barr positive. We have the greater representation of the seen tumors. And this is mainly based, TCGA, in European descent populations, right? So unfortunately, populations such as African Americans are not well represented here. Indigenous populations and also populations from Latin America, they are not represented here. So we don't know if the same distribution that we see in TCGA will hold for these other populations. As an example, our colleagues from Alaska, they reported 20% of EBV positive gastric cancer, and that is doubling the estimate that we see globally. So it will be very important to have additional representation of these high-risk populations. And I think that part of that problem that we don't see the US populations capture in these efforts, even though NCI is supporting many of these, is really the lack of biospecimens, right? We still do not have enough in terms of gastric cancer, perineoplastic lesions. So I would like to make a call for all of us to continue working hard on trying to improve and to have larger numbers of biospecimens. So the two questions that are presented today are, can non-invasive biomarkers be used to accurately detect gastric cancer? And here, I would like to emphasize that I am referring to early stage gastric cancer, because we don't need really any tests to diagnose gastric cancer in an advanced stage. And the second one is, can biomarkers be used to accurately determine which individuals are at risk? And I am particularly interested here in individuals with intestinal metaplasia. It will be wonderful if we will be able to develop a non-invasive panel that will tell us if someone is likely to have intestinal metaplasia, so we can then refer them to endoscopy, do the proper mapping, and classify the risk, right? That is the dream that many of us have. Just also to refer to, as Dr. Jay and Dr. Pesuelo mentioned, intestinal metaplasia is heterogeneous. So we have heterogeneity in the tumors, but we also have heterogeneity in the perineoplastic lesions. So it will be important to also assess that heterogeneity. Here, I just want to emphasize the phases of biomarker development. This is not an easy task. This is based on a paper that was published in 2001, and you can refer to the paper or also refer to the EDRN network that is led by NCI. So in general, there are 5 phases for biomarker development, right? So we start with observations comparing, for example, tumors, tissue, right? Malignant tissue versus non-malignant tissue. Then we move to cross-sectional comparisons of, for example, invasive cancer and controls. Then we move to pre-diagnostic samples, right? Samples collected prior to the development of cancer and compare that to controls. That's phase 3. So we can start with phase 1, or we can jump to phase 2 or 3. There is not a specific order in the first 3 phases. But then for phase 4, which is the application of something that we believe that will be useful, we need to do it in order. And then number 5 is really to see if these specific biomarkers will reduce mortality, which is finally the outcome to determine if a screening program really is useful. The Early Detection Research Network, EDRN, was established by NCI about more than 20 years ago. And this is a consortium that is devoted to discovery, validation, and basically development of biomarkers for early detection of all cancers. In these 20 years, and with the collaboration of many institutions and about 300 investigators, they have been able to develop 18 markers that have CLIA certification. Here I am just referring to 2 that are useful for us, and those are the ones that are associated with Barrett esophagus, because Barrett esophagus is basically metaplasia in the esophagus that, as I understand, is mainly incomplete type intestinal metaplasia, which is the one that we care in the stomach. So these 2 are CLIA certified and are being used, but the other ones that I am not mentioned here are related to particularly with lung cancer. So the process is difficult. And in terms of the stomach cancer, the domains that have been investigated are proteomics, microbiomics, genetics, epigenetics, metabolomics, and autoantibodies. I am referring to some of the more I would say key papers, and those who are at least in phase 3 using pre-diagnostic samples. So starting with pepsinogen, we know that pepsinogen has been widely used in Japan, as I understand. In some areas they are still use pepsinogen, but unfortunately it's still suboptimal. And I think that one of the reasons, besides the ones that were mentioned yesterday, is that potentially there is genetic differences in the expression of pepsinogen, right? This is a summary of one of the more recent meta-analyses for pepsinogen, 49 studies. And as you can see, this is for atrophic gastritis in the corpus. So the pool sensitivity 70% and the pool specificity 93%, area under the curve of 90%. In terms of gastric cancer, pepsinogens 69% sensitivity and 73% specificity. So they are not bad, but they are not good. In my opinion, this is the first step in the stair. So we need to do pepsinogen and then try to build for this multi-marker panel to push sensitivity. In terms of the inflammation-related biomarkers, we know that inflammation is the common denominator for stomach cancer. Here I am just highlighting the studies that have used nested case control studies. These are in pre-diagnostic samples, and also early stage gastric cancer. They are mainly addressing chemokines, interleukins, and other biomarkers associated with inflammation. And the message is that unfortunately they don't discriminate enough. Something that is coming that I think that will be very useful is proteomics. The CPTAC is an NCI initiative that is the Clinical Proteomic Tumor Analysis Consortium. So this is similar to TCGA. So they are doing proteomics in several types of cancers, and one of the current ones is stomach cancer. So for this particular analysis, they have about 165 gastric cancers and also 41 tissues from the adjacent non-malignant. And they are doing proteomics, the comprehensive platforms, and they have identified more than 12,000 protein groups, and the median per sample is about 10,000. So they are analyzing changes in protein expression, modification, pathways, and this effort really will move the field in terms of potential diagnosis, because something that we see in tissue we can then test in circulation, right, or in other biological matrices, and also in terms of personalized therapeutics. So this is coming, the analyses are ongoing, and it seems that the paper will be ready in about a year. So please keep tuned with that one. Moving to serology of H. pylori, this is a summary of the studies based on ELISA. We all know that all these estimates are completely underestimating if we assume that most of the non-cardiac cancers are associated with H. pylori. For samples that were collected less than 30 years of the outcome, these are mainly pre-diagnostic samples, so the relative risk is about 2.5. For samples that were collected more than 10 years, is a little higher, it's about a 4-fold increase, and the overall is about 3. For studies that have used a multiplex or immunoblot, we have that for samples that were collected less than 10 years, it's about 3, and for samples that were collected greater than 10 years, it's about 15. The overall is about 9. These provide the better estimates. And in terms of the specific antibodies to the various epitopes of H. pylori, Dr. Eplein summarized some of the results of the studies that she conducted. There are other here, and in general, the message is that the humoral response of H. pylori is really silent, right? We did a study using more than 1,500 epitopes, and we saw signals in about 50 of them. We need to remember that H. pylori has evolved with humans and wants to be, right, with humans. So just with the exception of some specific antigens, such as KG, we see strong responses, but still they are suboptimal. So this is, this is adding to the pepsinogen serology, and I completely agree with Dr. Eplein. KG will be a potentially a good discriminatory marker. In our analysis of the GCPL study that we are conducting in Chile in collaboration with Dr. Riquelme and also Dr. Alejandro Corbalán, we see that the association with intestinal metaplasia, particularly the advanced one, the odds ratio is about 10. So it's like we are building, but we need additional markers for more to increase sensitivity. Combining the ABC classification that is used in Japan, this is a beautiful study, almost including about 4,500 individuals followed for about 16 years. And here, I just want to emphasize the 2 points, that the ABC classification is useful not just for the intestinal type gastric cancer, but also for the diffuse type. We know that some of the diffuse type cancers are associated with H. pylori. Not all of them, but I think that at least 60% of them are associated. And as you can see here, for intestinal, we see for group C and D, we have that the odds ratio for C, for example, is about 24, and for D is 67. And for diffuse, group C, 11, and group D, 73. So clearly, if you combine pepsinogen with serology, you are increasing sensitivity, but we need something else to really be able to increase sensitivity. For Epstein-Barr positive gastric cancer, which is a very interesting area of research, and I want to encourage all of us here and also online to really assist with this one. This is a very interesting one. No, it's like Epstein-Barr is a very pleomorphic virus that is associated with no disease and associated with lymphomas and solid tumors. And let's remember that Epstein-Barr positive gastric cancer is the more common malignancy associated with Epstein-Barr. And we have this study, a collaboration with several collaborators, some of them here, Dr. Morgan. We have a signature for the non-invasive identification of Epstein-Barr positive gastric cancer. So we need additional validation for this, but we have results in the right direction, in the sense that some of the markers that we identify, we tested the full epitope, all the epitopes of the virus, and we see that some of the associations that we see, the magnitude is about 100. So we are in the right direction. We need additional validation. As you can see here, there are small sets, because in general, Epstein-Barr positive gastric cancer is rare. So we need to do more pooling and to try to understand this one. And this is important in the sense that individuals with Epstein-Barr positive gastric cancer are the ones who will benefit from immunotherapy, right? So it's important to really make additional efforts for this type of cancer. Now moving to genetic susceptibility. So this is a summary from the GWAS catalog. This is a site that you can visit to see the summary of the GWAS for any outcome. So this is for stomach cancer. I did this search in June of 2024, and this is combining about, I will say, 40,000 gastric cancer cases, and we are behind if we are compared to, for example, breast cancer. They are close to 300,000, so we are a little behind, and behind of colon and others. But we are moving in the right direction. These are mainly East Asian populations, which is is very important. We have some studies in Western populations. We are just finishing our GWAS in Hispanic populations with Dr. Morgan and others here. And we have also a GWAS in Western populations that will supplement the ones that are already published in European populations. Basically, as you can see, a relatively limited number of genes, about 100, if we compare to other cancers. And specific hits, I want to emphasize this one in chromosome 8, PSCA, that is a hit that was even seen in candidate gene analysis. These are all GWAS, and there are other signals. And these GWAS are now translating in polygenic race scores that are basically combination of significant hits. And the good news is that the one that was published by Jin's group from China potentially translate to Western populations. And we also tested that one in Hispanics, and it's translated. So the good news is that in terms of genetic susceptibility, potentially there will be, there is hope. And indeed, for the polygenic risk score, for individuals in the quintile, the fifth quintile versus the first one, the odds ratio is about 2. So if we can just also add some SNPs to pepsinogen and serology and others, I think that it will be good. We are trying to do, and we are moving in that direction, a GWAS of intestinal metaplasia, the goblet study. So if you have any samples, I would like to collaborate with us. We want to see if the hits that we are observing in gastric cancer are also valid for intestinal metaplasia. Moving to microRNAs, I will not delay. Dr. Jeff kindly presented the data in detail, and this is the GastroClear that was also presented by Dr. Hur, that is already being used in the clinic, and the additional microRNA study from Dr. Aba from Japan. So clearly microRNAs are useful. We know that they are working in Asian populations. There are some papers that suggest that expression of microRNAs are variable by race, ethnicity. So it will be interesting to see if these signals are also translate potentially to other populations, and that is an important point of consideration. So methylation. So methylation is really a good target for gastric cancer. This is a summary of the methylation for TCGA, and the ones in color represent the 4 groups of TCGA. And as you can see, Epstein-Barr positive gastric cancer is the so-called king of methylation of all tumors. And even more methylated than the original SIMP of colorectal cancer. That's very interesting. It's a completely different pattern. And the other 3 types of cancers, as you can see, they also have high expressions of methylation. And for example, SIN, that is also important for esophageal. And as I mentioned, there are 2 EDRN tests that are already available. They are based on methylation. So methylation is a good target. And the studies of Barrett, they are methylation in the tissue, either cells that were taken through like a sponge, or a paraffin-embedded tissue. So I think that methylation will be very interesting to study for stomach cancer, particularly with samples directly from the stomach. This is the multi-cancer early detection test, that is methylation in circulation. And as you can see, it was done for many cancers. We have stomach cancer here with a sensitivity of less than 75%. They don't provide a specific distribution for gastric cancer, but we know that some of these tumors are already advanced. So it is always important to include early stage, because that's really the key group to determine if these biomarkers will serve, and hopefully also preneoplastic lesions. In terms of sex hormones, I have been interested in sex hormones for a while. Gastric cancer predominates in males, but unfortunately we are seeing that the new trend of early onset gastric cancer is now moving to females, right? So it's like we haven't solved this one and we have another one. So the disease is evolving. Interesting to be in this field at this moment, right? So for hormones, these are the studies, and unfortunately nothing really meaningful. These are mainly estrogens, and we are expanding to androgen also, but basically the message is that potentially no. So we are trying to move to gastrointestinal hormones as a next target. This is a meta-analysis that summarizes proteins and metabolites. Here, for example, trefoil factor is not present here. Trefoil factor is an interesting one to study. We know that it's overexpressed in cancer, particularly invasive. There is a study also from Asia that Dr. Emura brought to my attention yesterday in combination with pepsinogen. So trefoil factor, we need more data on that particular one. We did a study in Chile, but unfortunately our sample size was limited. So I think that we were underpowered to see something for intestinal metaplasia. But if you have the opportunity, I think that is one of the ones that we should target. And here, so we see ferritin, transferrin as potential factors for this. Recommendations. So the recommendations are to collect and maintain informative biospecimens. We need absolutely more biospecimens, references that we can use, what kind of groups to target. Early stage gastric cancer, intestinal metaplasia, hereditary gastric cancer, and also pre-diagnostic samples. What kind of biospecimens? please try to collect everything, because I think that for potentially saliva I'm going to explain that in a second, will be good. So try to collect all of them, and you can, as described, jump to phase 3 if necessary, if there are compelling data suggesting that could work. And this is just to summarize ongoing collaboration. So we are doing a fragmentomics in saliva in collaboration with UCLA, so Dr. David Wong and Nettie here. So we are doing saliva because I think that it is important. Oral health is very important for all digestive tract, right? And I think that it will be very relevant, particularly in the absence of H. pylori, because as soon as you remove H. pylori from the stomach, the bacteria that we have in our mouth will try to colonize the stomach. So if we can develop something in saliva, it will be good. So, Nettie and Dr. Wong, they encouraged me to collect some saliva samples in the GCPL study in Chile. So we are going to start fragmentomics there. Extending on fragmentomics, which seems to be very more frequent in circulation. So this is a collaboration with Victor and Zach from Hopkins, and it's a multi-center study. We have some samples from the Americas, Europe, and Asia, but if you have samples, please let us know. They have some preliminary data suggesting that potentially that will be useful. And just to end, the EnteroTrack. EnteroTrack is the string test that has been used for decades. Dr. Correa, my former mentor, he used EnteroTrack and other colleagues to isolate H. pylori, particularly from indigenous populations. So with EnteroTrack, in partnership with Robin, who is the CEO of EnteroTrack, we are trying to see if we can do diagnosis of H. pylori. So I encourage him to try to do a urease test with the string, and in collaboration with Javier and also Bali from Peru, they have some experience doing urease with the string. He is now developing urease for the string, and it seems that it's working. We would like to do some omics with Han Lee. He kindly offered to do some of the genomics with the strings. We are going to do proteomics. It will be so beautiful if we just with the string, we will be able to know if someone is infected at this moment, and if we can potentially do some biomarkers there. One of my colleagues at NCI is thinking that even potentially H. pylori resistance could be also being done with the string. We want the string also for isolation of H. pylori from Africa, to expand our efforts of the H. pylori Genome Project. So the take-home messages are most biomarkers only exhibit low to moderate diagnostic value, so we need to combine, right? We need more organized and sustained efforts to move the field forward, right? Emerging technology are moving fast, but we are not prepared in terms of, we don't have the samples to do it, right? So we need to really do more efforts in terms of that. And this one is just in case that you miss the NCI think tank on advancing gastric cancer prevention. No, please, all the sessions are recorded, and we were very pleased to have more than 200 colleagues interested in the field, and we need to continue raising the gastric cancer awareness, and also try to have a more ambitious and more consistent and hopefully well-funded agenda that will help us to move the field forward. I think that we have waited too long, so I think that this is the time for that, and I again thank you to you and Robert for this protected space to discuss. Thank you so much.